Good day everyone. Today our group will be presenting about tumor necrosis factor receptor 1 signaling pathway, also known as TNFR1. This presentation will be presented by Tam Aisen, Fiza Kosar, Shreya Singh, Shana Nasreen, Alia Siad.
Today I'm here to discuss on the concept of tumor necrosis factor receptor 1. So this pathway is essential for our system's response as well as for handling the inflammation to keep us compact to illness. So we'll delve into the elements at play, the TNF itself along with its receptor, and the subsequent processes that occur. Initial step in the TNFR1 signaling pathway is when a TNF trimer binds to the extracellular domain of the TNFR1, leading to the release of an inhibitory protein. So the inhibitory protein is just a silencer of the death domain, which will be released from the extracellular domain of the TNFR1. So there will be a formation of plasma membrane bound complex, which is a complex 1, which consists of TNFR1, which is a receptor for initiating the cell signaling pathway, the adapter TRADD, which activates the pathway. So TRADD is a TNFR1 associated signal transducer, which is for activating the pathway. The kinase RIP1, which is a caspase independent pathway of programmed cell death, and finally the TRAF2, which is an intracellular adapter protein. So these will rapidly signal for the activation of the nuclear factor, kappa light chain enhancer bacteria B cells, or in short, NF-kappa B.
In the next step, it lacks TNFR1, TRADD, and RIP1 associate with FADD and caspase 8, which forms a cystoplasmic complex. Then, when NF-kappa B is activated by complex 1, complex holds the caspase 8 inhibitor FLIPL and the cell survives. The secondary complex is the one that initiates apoptosis if the NF-kappa B signal from complex 1 fails to be successful in expressing the anti-apoptotic proteins such as FLIPL. Now I'll let Aishan explain the function and importance.
Pause! I totally forgot to explain what TNF pathway even is.
Oh my god,
Who invited this
man.
group? Anyway, the tumor necrosis factor is a potent cytokine that exerts pleiotropic functions in immunity, inflammation, control of cell proliferation, differentiation, and apoptosis. Now back to the presentation.
I'll be discussing the consequences and approaches to address the dysfunction. There are 3 drugs which are currently licensed as TNF blocking agents, and they are Humira, Remicade, and Enbrel. They are used to treat rheumatoid arthritis and other inflammatory diseases, including ankylosing spondylitis and Crohn's disease. The US Food and Drug Administration has reported certain serious but uncommon adverse events with all 3 approved anti-TNF agents, including serious bacterial infection, tuberculosis, and certain opportunistic infections, demyelinating syndromes, and systematic lupus erythematosus-like reactions. In controlled studies of TNF blockade, more cases of lymphoma occurred among patients receiving the agents than among controlled patients. And in open-labeled uncontrolled studies, the rate of malignancies in the treatment group was several times higher than would be expected in the general population.
Now I'll be talking about functions and importance. The first function is that it will promote inflammation. TNF-alpha will bind to TNFR1, in which will form complex 1. This complex will lead to the activation of LKB kinase, which will phosphorylate LKB. And by phosphorylating LKB, it will cause its degradation, which allow NFKB to translocate to the nucleus. And once in the nucleus, NFKB will induce the expression of pro-inflammatory cytokines, chemokines, and adhesion molecules that drive inflammation. Not only the complex 1 will activate LKB kinase, complex 1 will also activate MAPK. MAPK will regulate the expression of inflammatory mediators and contribute to the production of cytokines and chemokines. The second function of TNFR1 is cell survival. By this function, it will prevent apoptosis, which is cell death. And by preventing this, it will allow cells to resist death signals and continue to function under stress conditions, like during inflammation.
The third function of TNFR1 is to promote the expression of genes involved in the cell cycle progression. This occurs when the tumor necrosis factor receptor binds to transmembrane tumor necrosis factor (TMTNF), initiating signaling and forming the TNF-TNFR1 complexes. Complexes ILA and ILB can induce classical apoptosis, with capsase 8 as the main executioner. Meanwhile, complex ILC drives necroptosis via receptor interacting serine and threonine protein kinase 3. Now, the fourth function of TNFR1 is the transition to complexes ILA and ILB, which induce apoptosis. This process involves several key events. Chromosomal DNA fragmentation occurs, cytoskeleton protein degrades, and membrane bulbing forms on the cell surface. As a result, the cell undergoes shrinkage and detach from the neighboring cells. Lastly, the fifth function of TNFR1 is the transition to complex signaling in innate lymphoid cells ILCS, which triggers necroptosis. Necroptosis occurs when pathogen or stressors block apoptosis, ensuring the elimination of infected, damaged, or stressed cells. This process contributes to tissue's integrity, immune defense, and the resolution of inflammation. By helping to clear infections, necroptosis play a crucial role in maintaining immune balance, particularly at the mucosal surface like the gut.